Assalamu alaikum. Welcome to EP6 online support program. This is class 10th, week 11, lesson 1 and subject biology. Textbook biology, page 138 to 140, unit 17 biotechnology. Topic is introduction to biotechnology and fermentation. Objectives. By the end of the lesson, you will be able to discuss old and new biotechnology, fermentation and its types. Introduction of the biotechnology. So the biotechnology can be defined as the use of microorganisms for the manufacturing of the useful products for the welfare of the mankind. Basically, the term biotechnology is new in biology, but as a discipline itself, it is an old term. For example, the artificial breeding in animals and the cross-pollination in plants, these are all the old techniques, as well as fermentation is also an old technique. So all these are includes in old biotechnology. So we can classify biotechnology as old biotechnology and new biotechnology. So the new biotechnology is basically known as genetic engineering, in which we can easily play with the genetic material, that is the DNA. So we can simply classify the biotechnology as old biotechnology and new biotechnology or modern biotechnology, or it is also known as genetic engineering. Modern biotechnology. Now we will define the modern biotechnology, that is also known as genetic engineering. That is artificial synthesis, modification, addition and repair of the genetic material, that is the DNA. This is considered as the modern biotechnology. Why all these processes happens? Why the modification, addition, cutting, repairing, pasting and synthesis of genetic material done? Because to change the characteristics of an organism, if we need to get an organism with our desired characteristics, so we can change their DNA easily through the modern biotechnology. So in 1944, the proper working on the genetic engineering starts, when it was proved that yes, the DNA carries all the genetic information of an organism, so the scientists start working on the genetic engineering. In 1970, the scientists were able to cut and paste the DNA of the organisms. If we want to change some characteristic of an organism, or if an organism has some defect, some genetic disorder, so we can easily cut that part of the DNA and paste it with our desired gene or desired characteristic. So in 1970, this cutting and pasting of the DNA starts. In 1978, the human insulin gene was prepared. As we all know that insulin is really an important hormone of the human body, as it controls the sugar level in the blood. So this insulin gene was artificially synthesized inside the E. coli bacteria. So in 1978, this insulin was prepared. Human growth hormone was also synthesized in E. coli bacteria. In 1990, the Human Genome Project was launched. Genome means the genetic material. That, as we all know, that human has 46 number of chromosomes. So which chromosome has which type of gene and which gene is located on which chromosome? So all these study was started in 1990 and this was completed in 2002. So it get almost 12 years to complete or to study the Human Genome Project completely. So by the help of the Human Genome Project, we get to know that which gene is present on which number of chromosome. So this was all about an intro of the modern biotechnology. Fermentation. The fermentation is also known as anaerobic respiration. So now we will define the fermentation as the incomplete oxidation reduction of glucose is known as fermentation. As it is anaerobic respiration, so that's why the glucose is incompletely go through the oxidation reduction reaction. At first, this process was thought to be a pure chemical reaction because at that time it was not believed that the microorganisms are involved in fermentation. Then, in 1857, a scientist known as Louis Pasteur explained that specific fermentation is carried by specific microorganisms. So it means we have different types of fermentation. That is, the fermentation can be classified in terms of product formation. Like we have alcoholic fermentation, like we have lactic acid fermentation. So these are the different types of fermentation which can be carried out by the different types of microorganisms, which will be discussed later. This is a flow sheet diagram of carbohydrate fermentation in which the glucose is broken down into pyruvic acid, which can be further converted into alcohol, that is ethanol, lactic acid, and other organic acids. Now we will discuss the different types of carbohydrate fermentation. So the first one is alcoholic fermentation by yeast. As the name indicates, in this process the alcohol, that is the ethanol, will be formed. So this type of fermentation is carried out by a yeast, which is basically a unicellular fungus, and it is also known as Saccharomyces cerevisiae. In this process, the pyruvic acid is converted into alcohol, but before conversion into the ethanol, the pyruvic acid is first reduced to acetaldehyde. Then the acetaldehyde is reduced to the alcohol. Basically, the acetaldehyde is the intermediate step between the pyruvic acid and the alcohol formation. The alcohol formed is known as the ethanol, which is a 2-carbon molecule, but we know that the pyruvic acid is 3-carbon, so it means 1 carbon is released during the conversion of pyruvic acid into the ethanol. The carbon release here is of carbon dioxide, so the carbon dioxide form is used for the rise of the bread. This process can also be used for the wine formation. This diagram shows the alcoholic fermentation, in which you see the pyruvic acid is first reduced to acetaldehyde, which is then converted into ethanol. In the meantime, the CO2 is released, which is then used for the rise of bread, etc. The second type of carbohydrate fermentation is lactic acid fermentation by bacteria. As the name indicates, in this fermentation, the lactic acid will be formed by the help of the bacteria. So the Streptococcus and the Lactobacillus species of bacteria are responsible for the formation of the lactic acid, and this process is really important in dairy industry for the souring milk. Souring milk is used in baking, etc., and for the production of cheese and yogurt. So the pyruvic acid is reduced to the lactic acid by the help of the Streptococcus and the Lactobacillus bacteria. The lactic acid is found in milk.